My name is Bruce Stillman. I'm president of Cold Spring Harbor Laboratory, and welcome to the lab. So my job is to introduce today's speaker, who is Michael Wigler, who's been here at the laboratory for slightly longer than I have, which is well over 30 years. Mike was not atypical, but was one of the first scientists that Jim Watson hired straight out of graduate school when he finished graduate school at Columbia University. While he was a graduate student, he developed one of the most important techniques that has been used in the biotechnology industry that has led to the development of drugs like recombinant insulin, human growth hormone, the drugs to treat side effects of cancer, Epogen and Neupogen, and many, many other biological drugs that are made by companies such as Genentech, Biogen, and others, Amgen and others. Without this technology, none of those companies probably would have existed. And so this was a really very, very important discovery, and Mike did that when he was a graduate student. He came to Cold Spring Harbor, continued on that work, and I think the first important and big splash that he made at Cold Spring Harbor, which was a few years later, where he isolated the first cancer gene out of a human tumor. And then spent the rest of that decade working on cancer genetics and is a pioneer in the field of cancer genetics, which he's going to talk about. But then Mike continued to push the envelope and develop other really important technologies that many of us use in the laboratories. And when the prospects of sequencing the human genome were at least initially discussed, Mike was one of the first people not to get involved in how to sequence the human genome, but started thinking about what are we going to do once we have the human genome complete. And he has developed very important technologies that led to his interest in studying diseases like autism and other neurological disorders, and continuing his studies on cancer. He was then soon later joined by Jim Hicks, who's here as well, they have a major effort on breast cancer, which Mike is going to talk about. But Mike has been described to me by other scientists as being a scientist. He's the type of scientist that other people go to seek advice and seek ideas for their projects, and he's had an enormous impact in science and also at Cold Spring Harbor Laboratory, and I want to thank Mike for giving today's address to you on breast cancer. Mike Wigler.
Those are very kind words. I hope Bruce won't regret having asked me to give this talk. I came to the lab when I was a graduate student because, primarily because spectacular work was done at Cold Spring Harbor, and the science that came out of the lab was an amazing thing. But also it had a tradition of eccentric scientists, and I felt that I would be at home if I came here. And I was right on both counts, and so I came here after graduate school, and although I was been tempted by various places to leave, I never have. So I'm going to try to stay on the subject, but I probably won't be terribly successful. People have asked me what I was going to talk about, and I said, well, I don't really know, because typically I speak extemporaneously. But on the way over, I thought of a joke for this occasion. I spoke with Bruce about whether this joke was appropriate, and he said it isn't, but then I checked with my wife, and she said it was okay. And then, as you'll see in a moment, I also checked with somebody else about whether this was officially a fundraiser. Bruce said it wasn't a fundraiser, and that half of the people here were new, so I shouldn't really chase them away. And but somebody else I spoke to said, yes, it's definitely a fundraiser because there's a raffle. Okay, so here's the joke, and I'm going to tell you the punchline very quickly, because if you think about the joke, you'll come up with the punchline yourself. and is, what is it that sex and a fundraiser have in common? And the answer is that you can have fun while giving joy to others. Okay. Don't transpose any of those words, or you'll come up with another joke. All right, so the title of my talk is apparently, I brought this thing with me, Fighting Cancer One Cell at a Time. Now, this title came from discussions with Charlie Pritzi, because my original title was a little bit too obscure, and he wanted something with more punch to it. Fighting cancer one cell at a time sounds like, it sounds like fighting words. In fact, I'm going to be talking about looking at cancer one cell at a time, but not from the point of view of fighting it with therapies, which is what Bruce has talked about and what Bruce and Jim have both championed, but catching it very, very early on, at the earliest possible stage, before the cancer has accumulated so many mutations that it is really an uphill battle. And so I'm going to summarize my talk in a single sentence, and then I'm going to explain from an historical perspective how we came to be at the point we're at. So the summary of the talk is that in a few years' time, perhaps 2 to 5 years, we'll be able to walk into a doctor's office, he'll draw a blood sample, and there'll be a fairly routine and inexpensive test that tells you if you have cancer somewhere and in what part of your body. So that's the talk. So how did we get to this? Well, obviously, one part is technology, and another part is conceptual. And another part is, so half is technology, half is conceptual, and half, the proverbial other half, is experimental observation. We've been involved in all three of those, and so now, hopefully without boring you too much, I'm going to give the history of that, because I think understanding science from an historical point of view is the right way to understand science. When I came to the lab in 1978, there was already the work by Bishop and Varmus, for which they later received Nobel Prize, that viruses contained genes that could cause cancer. And those genes were actually genes that had been stolen from our own genomes, that the viruses had mutated and had exploited to the advantage of the virus and caused disease in animals. So we were already primed with the notion that cancers might be caused by mutation of genes that would be by their mutation, either by their loss or by gaining of some new property, could cause cancer. And at the urging of Jim Watson, we utilized the technique that I developed as a graduate student to search for these oncogenes, and Bruce spoke about that. We were the first team to isolate. We were first by, I think, about a week, which doesn't mean anything. But anyway, we were the first by a matter of maybe a week to isolate the first human gene from human cancer that we could demonstrate by putting this into a normal cell, could convert that normal cell into a cancer cell. And that began many decades of very productive cancer biology throughout the world, not because of our work, but because the time was right, to begin to isolate cancer genes, what we call cancer genes. And these were isolated by many different methods. Some of them used our methods, others used chromosome translocations, variety of techniques to discover things that we now call oncogenes. These are genes that, when activated, can cause cancer, and also genes that we now call tumor suppressors. These are genes that prevent us from having cancer, and if those genes are lost, then you can be prone to cancer. One of the most famous of these, of course, is the BRCA1 gene, which was identified by Mary Claire King using population genetics technologies and some molecular cloning by other people. I identified this gene. And the knowledge of this gene and being able to diagnose it in families has been one of the milestones of cancer research. So these genes, tumor suppressors and oncogenes, are in some sense the keys to understanding human cancer. The methodology for discovering them soon became, when I say soon, I mean about 14 years after the discovery of the first one, became the obsession of my laboratory. And we went through a series of technical improvements in methods that are called genomic difference analysis, where you take a cancer cell and you take a normal cell and you compare them and you ask what's different about the cancer than the normal cell, and those differences might be responsible for the cancer. So we worked out methods for doing that. Those methods led to a discovery of new cancer genes, tumor suppressor genes like P10, which is frequently involved in breast cancer and prostate cancer, but also to some viruses. That technology began to lift the cover on the ways in which our genome becomes rewired in a cancer cell. And the technologies got stronger and stronger, and when it was clear that the human genome was going to be sequenced, we were really ready to exploit that information. And at around that time, Jim Hicks, who's here, where's Jim? Where? Oh, Jim, hi. Jim rejoined Cold Spring Harbor. Now, Jim came to the lab, I think, a little bit before I did, was a yeast geneticist and had worked on what might seem like an obscure problem. He worked on how yeast changed their sex. So sex enters the conversation again. But yeast flip-flop. They come in two sexual varieties and they switch back and forth. And Jim figured out, together with his team, how they do that. That was very hard to top. So Jim left the lab and ended up in industry and worked for many years in industry, but he was drawn back to research, and I was fortunate that Jim decided to do a sabbatical, which was going to be a brief sabbatical in my lab to learn this technology, and he stayed. That was in 2002 or so, 2003. So Jim and I worked on further developing this technology to really decode the book, the cancer book. And we published in 2006, I think it appeared in 2006, sort of the manifest, if you like, of many breast cancers. And what we saw really was somewhat shocking, although in retrospect, perhaps it shouldn't have been so shocking. And that is that cancer cells contained dozens upon dozens of mutations, many of them recurrent, so that would suggest that they're causative. But there were far too many for us to try to make really a clear rifle shot. There was clearly multiple causes, even in a disease as you would think as unified as breast cancer, the causes were going to be too many. And we published this paper, and it forced us to take stock of what we had seen. So you have to understand that going into this, we were expecting to find the cancer genes, and it's a little bit like a kid who goes to fight some bully and then realizes the bully's got bullies behind him and decides maybe it's better not to pick that fight. So it seemed to us at that time that perhaps what we could best do is use these methods to help in diagnosis of cancer, that is, understanding which cancers were lethal, which cancers would respond to which drugs, and that would be a great assist to the oncologist. So Jim began several efforts in that direction, while I retreated back to the bench together with a graduate student named Nick Navin to work on developing even more powerful technology, that is to be able to do what we did with cancers, but from a single cell. Now, often, the advances that occur, at least in my life, have occurred on the shoulders of other major achievements. So the achievement that led to discovery of the oncogene really rested on the shoulders of what's called now the recombinant DNA revolution, which came out of bacteriology, people studying little pieces of DNA that could serve as their own little chromosomes, and you could use those DNAs to transfer back and forth between cells. So the early part of my career got an enormous boost from the recombinant DNA technology. Then at another point, around the time that we developed precursors of our current methodology, there was the development of the microarray. And some of the people in this room helped support us in that effort. And this was largely a revolution in the synthesis of DNA and the fabrication of small devices. And then further on, in the most recent phase, we've been helped by, as I already mentioned, cloning the human genome, sequencing the human genome, so that we had all the building blocks at our disposal. We could tell what was normal and what was not. And in this most recent chapter rides on the shoulder of DNA sequencing technology, which is the advent of machines that are orders of magnitude more efficient than the existing technology for sequencing DNA. These have produced a revolution in biology that has exceeded, I think, the revolution that was caused by recombinant DNA. As a result of that technology, we now can talk about sequencing from a single cell and getting the contents of a single cell. And I'll talk to you in a moment about what that means. I'm just going to take a slight digression because I feel like it, which is, in fact, I'll take a digression on the digression. It's 2012, so about 12 years ago, Jim and Bruce showed me the plans for the buildings on the hill, which I'm sure you've all seen. I thought it was really a dreadful idea because we didn't know if funding in science was going to keep pace, and it just seemed like a distraction for the lab at a time when the money raised should go for research. So I thought that was a horrible idea. And then our lab was the first to move in. Now, I didn't want to move in. And the way I think, my understanding of this is that what Bruce did was he went to the people in my lab and took them on a tour, and they came back and said, "We're moving." And then a month or so after moving in, which was about 2 or 3 years ago, I said to one of the people, do you want to move back? Shall we move back? And he said, "Over my dead body." So it actually turned out to be very good that moving into new quarters with a view of the harbor was a very beneficial thing. So they were wise in that respect. That's the digression on the digression. So now I'll get to the main digression. So about, I guess it was 4 or 5 years ago, Jim got a large gift of money from the Stanley Foundation. I don't remember how much. It was really an extremely nice gift. And came to tell me about it, and that what he was going to do was to buy sequencing machines. And I said, well, that's a horrible idea, because there's nobody in the lab to even interpret the data that these things are going to turn out. They turn out so much data that nobody could make any sense out of it. And of course, Jim was right. And the lab then built the Woodbury Genome Center and equipped it with these spectacular machines, and now my lab is one of the primary users of it. So anyway, so then we're talking about these machines. And yeah, you should give an applause to Jim for... So now I'm going to continue on this same theme, but this is no longer a digression. So Jim and Bruce both think that are optimistic about treating cancer. And again, I'm somewhat pessimistic about this, but they'll probably be right. But in the meantime, it'd be great if we didn't have to get cancer. And so now I'll return to our work. So what does being able to sequence single cell mean? Am I actually obliged to talk for an hour? No, okay. Okay, good. So let me just take you through a thought experiment. So a thought experiment is, of course, was popularized by Albert Einstein when he was thinking about general relativity. But what a thought experiment does is it enables you to examine what your assumptions are and to see them clearly. So let's do a thought experiment. And you'll either learn something or well, you'll learn something. So the cancer genome has lots of mutations, and you can explore the cancer genome by extracting DNA and comparing that DNA to the normal DNA of the person, and then get that, what I call the manifest, or the inventory of problems with the cancer. Each cell contains only 2 copies, in general, only 2 copies of each gene, and the mutated gene may be present in a single copy, 1 molecule of DNA. So what single-cell sequencing means is that from 1 cell in that cancer, you can sequence that DNA, and from that cell alone infer a lot about what that cancer, the mutations occurring in that cancer, from a single cell. This is an amazing thing. I think we were the first to do it in cancer, but we weren't the first to conceive of this project. People had already been trying to sequence single cells from bacteria. It was the bravery of a graduate student, Nick Navin, with the urging of Jim Hicks and I, to try this in cancer, and he succeeded fairly quickly and found that the single cells, by and large, reflect the genome of the cancer. They might not have. The cancer might have been a total zoo of different mutations, and you couldn't capture that in a single cell. Well, it turns out that the cancer is a menagerie. It does have sometimes multiple rogue cells in it, and if you capture enough single cells, you can get the rogues gallery of problems in the cancer. This has lots of application. I'm going to just describe those applications, and then I will ask if you have questions. So the simplest application, the one that's fairly trivial to do, is often when we take biopsies, we take fairly large chunks of tumor material from the patient, particularly in the case of biopsies of the prostate. These can be, have high morbidity. There's the possibility that taking the biopsy itself causes inflammation, which could aggravate the cancer. It should now be possible to take really tiny biopsies from a suspected lesion, which introduces very little damage to the patient, from which, by doing single-cell analysis, we can determine whether the cancer is whether there is a cancer, and whether it's dangerous, whether the patient even needs surgery, or if they, even before surgery, should begin chemotherapy. That's an obvious application. We'll be seeing that application in the order of probably a year. The other applications are to be able to detect cancer at extremely early stages, and I'll start first with a case which certainly would have application. That is for somebody who has already had a cancer. The cancer has been removed by the surgeon, and now the patient, as a result of chemotherapy, seems to be in remission. It turns out that metastatic cancer is not quite as lethal as people had previously thought, that when metastatic cancer can be detected early enough, and there are only a few metastatic lesions, they are often treatable by surgery. The problem with metastatic disease is that we largely bury our heads in the sand after we've gone through this procedure, and we hope that everything is okay. And then when the body begins to fail, then you go in for tests, and oh my gosh, there are lots of bone metastases, there are metastases to the liver, and it's too late. It is likely, and this isn't our work, but it is likely that prior to that happening, there are cancer cells disseminated in the blood. And people had noticed this for a while, that there were cells in the blood that were abnormal, but they couldn't tell if these were cancer cells for sure. Due to experiments that Jim did, we now know that you can identify these cells as cancer cells, and that they have the genome of the cancer from which they derived. And this should provide a very early test for people that have been under treatment for cancer and are in remission, with the hope is that one can find the cancer recurring in the person at a very early stage. Now, these are, I would put almost in the bank as things that we'll be able to do. The most radical thing is, and it depends to some extent on forces beyond our control, would be to detect these cells before even the primary cancer has been diagnosed. That is, to find cancer cells in the bloodstream before the cancer has metastasized, and before the patient has a lesion that's palpable, or before there are physical signs. For example, many cancers, pancreatic cancer, ovarian cancer, you don't detect, you don't diagnose until it's too late. These cancers probably do shed cells into the bloodstream at a stage earlier than would be apparent on physical exam. And we're very hopeful that we'd be able to detect these by analysis of the cell. Analysis of the genome could tell us this is a cancer, and analysis of something called the RNA could tell us what tissue it's coming from. So that would be great, and that's our hope. It's sort of one of the main aims of the lab, is to demonstrate that this is feasible and to help facilitate it becoming a widespread technology. And we hope that this technology will be in the clinic within all of our lifetimes. Let's see if I've said everything I want to say. We could talk about anything else, but we'll leave it at that. Thank you.